Protocols and tips about plasmid transfection, RNA isolation, qPCR, and data processing by cyan. Declaration: this video is not an advertisement for reagents and equipment. Please use genuine software. All the cells mentioned in this chapter are from mammals. Firstly, I'd like to reiterate about the biological hazard. These organic solvents used in RNA extraction and the fluorescent dyes and radioactive dyes used in PCR are all toxic. Protect yourselves. We will start with the definition of a plasmid. A plasmid is a small extrachromosomal DNA molecule within a cell and can replicate independently. In its simplest form, a plasmid must contain the origin of replication. It usually contains selectable marker, and other elements are selectable. Here are the reagents you need for plasmid transfection using lipofectamine. When the seed cells are 70% confluent, dilute DNA plasmid and lipofectamine with optimum, mix them, and incubate for 10 minutes at room temperature. Add the DNA lipid complex to the cells. According to the protocol of lipofectamine, medium with serum and antibiotics is acceptable, and you don't need to change medium during the incubation thereafter. I tried and it went well. Sometimes the cells are vulnerable and sensitive to the toxicity of the ribosome-mediated transfection. You can use medium without antibiotics during DNA lipid incubation with optimum for 6 hours and then change back to the complete medium. Sometimes serum starvation before transfection and serum-free medium during transfection helps to improve the transfection efficiency. Polymerase chain reaction is a molecular biology technique for enzymatically replicating DNA without using a living organism. 3 steps are included in the PCR procedure. They are called denaturation, annealing, and extension. Each cycle doubles the DNA copies. There are some simple ways to increase the yield of PCR at the cost of miscellaneous bands, and vice versa. You just cannot have a cake and eat it. Here are the reagents we need for RNA isolation. The sample goes through lysis, chloroform extraction, isopropanol precipitation, ethanol washing, and then we dissolve them with the RNA-free water. Always keep your samples cool. It helps to slow down the RNA degradation. Be careful when you transfer your supernatant because it's much worse to have protein contaminations in your RNA samples as it helps to degradation. Use a bioreader to measure RNA concentration and then perform a reverse transcription. Here are 2 systems for different well plates using cybergreen as fluorescent dyes. The cDNA amplification goes through 4 periods called baseline, exponential, linear, and plateau period. The intensity of the fluorescent above background level, which is called CT value, is used to quantitate the newly generated double-strand DNA. We get the data from the software of light cycler like this. Copy it to an Excel sheet, just like the order we add the samples to the plate. As you can see, I mentioned the cell type I used in the upper left corner. 3 groups are listed as normal, vector, and P53 in the first row. Vectors means I added the transfection reagent and negative control plasmid, and P53 represents some kind of gene overexpression plasmid I used. Now, let's get started to the calculation. First, calculate the average of your internal reference. And the average of your target gene. Use this to minus this. You usually get a negative number because most housekeeping genes expresses a lot. Then use the power function. Get the average of your normal control group and divide the last row by it. Calculate the average of each group. The first group must be 1 because you just compare it with itself. The average of each group represents the relative expression of your target gene. In this example, negative control plasmid slightly upregulated the p53 and the overexpression plasmid successfully increased the expression of p53. If there are more samples in one group and their distributions are tested, you can calculate the p-value in this way. Statistical correlation is not causation. Do this to the remaining groups and you will see the same trend in each group. Now let's go on to the GraphPad to make a histogram. We use this row of data. I will simplify it to 1.43, 1.49, 113, and 79. Create a new column table, enter group names, and enter these data. Now we use a two-tailed t-test to see if it shows significant differences. Although it has rather large 95% confidence interval, p-value is less than 0.05 and we can mark it with an asterisk. Now let's move to the graph drawings. Add titles, and you can split the y-axis like this. Or you can always adjust x-axis, change colors and show data. Finally, create layouts and output it. You can find supplementary reading materials on my book. It includes other vectors to manipulate gene expression other than plasmid, and the truth of drug-resistant genes, and something beyond that. Thanks for watching. If you enjoyed this video, just replay it. If you have any questions or suggestions, feel free to contact me through email. If there is the next episode, see you then!